focus on this concept of neuroarchaealization. And this is something that we have going on in the lab for several years now. And very discrete research. I think most of my research is known for modeling diseases using stem cells. But yes, indeed, we do have like a line of research that focus on brain evolution. And I will start by saying that our biggest question is really to find what makes us humans. And that's not a simple task. And there are many ways to study that, and we bring the stem cell perspective on the table. So I think most people know that currently understanding of human evolution has indicated that many hominin lineages appear and coexisted on Earth. But only one, us, modern humans, survived to the present. So Neanderthals and Denisovans, two of these extinct lineages, are our closest relative. And we see them as the species that provide the most subtle genetic and phenotypic contrast to modern humans. And that's why we like to focus on them. Particularly, I would highlight the Neanderthals because these are the ones that we have the most information from the fossil records. So we know a little bit more about them. And one thing that it's really important in this context is this idea that throughout evolution, the Neanderthals, similar to any other species, didn't show the dramatic development in arts, technology, and adaptation that we see with modern humans. In a very short period of time, we, modern humans, we have dramatically changed the environment and developed technologies that are just amazing, not to be compared to any other species. So how actually this happens? What makes us a unique species in that sense? So there's many ways to start comparing the extinct relatives to modern humans. We take advantage of fossil records, especially for brain evolution. We look into endocasts. We can extract now ancient DNA from these fossil records. So we have genomic information from them. And there are also impressions that these extinct relatives have left. And we can try to understand them as well. So all of that give us like a view of how they live, where they have gone. I mean, how their societies were structured. But unfortunately, we don't have that much information about brain development, and that's because the brain do not fossilize. So that's where stem cell plays a role, and that's why we like to bring stem cell into the studies of evolution, because we can capture a time where no other model can actually give us too many information. So we approach that by using stem cells and differentiate these pluripotent stem cells into brain tissues, such as brain organoids, which is the topic for today. Before I continue, just to give like an overview of brain organoids, this is a technology that was initially developed by Yoshiki Sasai in Japan. And he was really the first one who showed us that if you grow these pluripotent stem cells and neuralize them in suspension, they will self-aggregate and form organized tissues that we never seen in our traditional two-dimensional cultures before. So these, he can even guide specific brain regions by adding ectopic factors in the culture media. So some of the images of his pioneer work are here, and you can see some level of organization, which is truly amazing that this was done in 2008. And that's exactly when I was starting my lab. So by looking at this technology, I thought, wow, this is something that I want to incorporate in my lab. So we start mimicking Sasai protocols very early on. But it was not easy. And my lab and other labs, were evolving these protocols over time. Many people have made adjustments to the protocol. We have much more robust protocols now and different subtypes of protocols as well. So when we talk about the brain organoid, it's important to define what kind of protocol we are using. And most of the time, we can classify brain organoids into two types. We call unguided differentiation. This was pioneer work from the Jurgen's lab with Madeleine Lancaster, where you have different brain regions in the same unit. or the guided differentiation pioneered by the Sassai lab, where you can use ectopic factors to drive specific brain regions. You can even combine different brain region organoids to create small circuitries in a dish. So each one of these technology generates a different type of organoid with different cell populations, different structures, and there is advantages and disadvantages for each one of them. So you really need to understand what kind of protocol you have to answer your biological question.